Alright then lads, this one is part 2 of amino acids. Now I don't know why I keep saying this, but if you haven't seen part 1, then you're going to have to go and watch that, because why would you be watching a part 2 without part 1, right? That's like watching Spider-Man 2 without seeing the first one. You know, you're going to be watching the movie and asking, why the hell is this guy spewing webs? Why has this guy got a red costume, right? It's a bit weird. So watch the first one and then watch this one. But for those of you who are normal, this is part 2. If you're new here, make sure to subscribe to the video, like, all that bollocks, and let's get this one done. So a quick last episode anime recap, right? Level 1 is the primary structure, and it looks a bit like this, okay? It's a very simple, regular structure. It's not very weird or complicated. It's just very straight chain, normal structure, okay? And then here you got the secondary structure, which is level 2. And here it starts to get all weird and mucky, right? It starts to bend and form these weird bonds in between the chain, right? Now let's talk about level 3. Now level 3 is where it really gets a bit crazy, okay? And we call this the tertiary structure, which just means 3, okay? Bit lazy, but you know. So here we have the secondary structure, okay? And let's just spam some more amino acids onto this big bad boy, okay? We just keep adding more and more into there, keep throwing it at him like a, you know, like a spell or something, just spamming it at him, right? We throw more and more at him. And of course, incidentally, it starts to grow larger and larger, okay? And that's no surprise, it gets bigger. Of course, so many more of these amino acids start to grow and get bigger. More chains kind of start to form attractions to each other. It gets even more muckier and messier, okay? You can see all these little bonds forming. And this is what we call the final 3D structure, okay? The tertiary structure is kind of where it ends, really, right? And I'm sure this is already apparent, but the thing is about this, and you need to really learn this, is that it is still a single polypeptide chain, meaning it is still all connected to each other, okay? It's not like you've just added another polypeptide, right? It's just this massive, massive chain, but it's all connected to each other. All 3 of them are all single polypeptide chains. Think of it as like a really long train, okay? A really long train has so many different trams connected to it, right? So many different trams, and it's still the same train. It's still connected via this long chain. Now here's where the tertiary structure starts to get all messy and mucky, okay? Now, these guys could contain both a alpha helix and a beta pleated sheet, right? They could have both best of both worlds. Both of best world. What did I just say? Oh god, my English ain't Englishing. Alright, let's continue. Now, of course, we already know that these little amino acids do cheat on each other, and they start to form bonds between other amino acids in the same chain, okay? And there's different types of bondings that you need to learn, okay? And we're going to go through that. So the first type of bonding you can get is hydrogen bonding, and we've gone through this before, okay? And this typically happens with hydroxyls, okay? These are very electronegative molecules. They have a slightly positive region on the hydrogen, and they have a slightly negative region of the oxygen. They're like biggest ops, okay? And when you put these two guys together, they form an attraction, and that's what we call a hydrogen bond. It's kind of like me when I see your mama, right? There's a deep attraction there. Now, these bonds alone aren't super strong, but when they start to grow in quantity, right, there's a large proportion of these hydrogen bonds. That's when it starts to get decently strong, okay? Not super strong, because it can still be broken by high temperatures and deviations in pH levels, so, you know, acidic or alkali. But basically, yeah, they're just, you know, a regular build. Now here we've got the second type of bonding which can happen, which is an ionic bond, okay? So let's say we take, I don't know, an amine, okay? An amine in place of an R group, okay? And this amine is, you know, positively charged. And let's say we take a carboxylate group, okay? And by the way, you don't really need to remember these names. So this carboxylate ion is negatively charged. And if you put these two bad boys together, you can probably guess what they do. Kind of like me and the Latina shorties, right? We make love, okay? There's a massive attraction here between the carboxylate negatively charged ion and the positively charged amine, okay? And again, you don't need to remember these names. Just understand that negative and positive, they attract each other. When you have 2 amino acids with their R groups being ions and they are attracted to each other, that's when you get ionic bonding. And this is stronger than your hydrogen bonding because ions are like very charged, whereas your hydrogen bonding is kind of charged, if you get what I'm trying to say here, okay? So this ionic bonding is a bit stronger, but the problem still lies in the fact that it can be destroyed with high temperatures and acids, okay? This acid here, this proton, okay? This is what we call an acid. This acid here can just come in and muck everything up, kind of like a cock blocker. And that's kind of the main reason why acid is so dangerous to you. That's why it destroys you and it just kind of eats away at your flesh. It's because it comes in and kind of just cock blocks the attraction between the ionic bonding and the hydrogen bonding and all these different attractions. It gets in the way and it starts to destroy these proteins, or a better word for it is that it starts to denature these proteins, okay? And what that means is when a protein is denatured, that means it no longer serves purpose, okay? It no longer functions correctly. And that's really bad, because then you just dissolve like a snowman, right? Like, I don't want to dissolve, and you don't want to dissolve either, right? Like, dissolving is, it's not very nice. So don't dissolve. Now, here's another type of bonding we have, okay? Now, this bonding is called covalent bonding. Now, this one is the big dog, right? This one, he means business, okay? Like, when he takes your girl, he really takes her, okay? You're not getting her back. This is actually an example you need to learn, okay? So here we have a sulfur atom, okay? And you put another amino acid with the R group as a sulfur atom. You put these two bad boys together, and they form a covalent bond. And we call this a disulfide bond. Now, this bad boy is really strong, right? He is him. When he takes your girl, she's staying with him, okay? You just forget about that girl and you move on. And usually when you find a protein with a high proportion of these bonds consisting of these disulfide bonds, those proteins are the things you find in your nails and in your hair. And that's the thing, they're structural proteins, right? They're made to be very strong, right? That's why your nails are pretty strong and your hair is really strong, is because they consist of these disulfide bonds that are built for structure, right? They're built different, and that's due to these covalent bonds. And these guys are made to be resistant to, high temperatures and, pH. They're made to be resistant against pressure. So, yeah, pretty cool, pretty awesome. Let's move on. Now, here we've got level 4. Now, I know I did kind of lie and say level 3 was the last one, but it can keep going, because level 3 is the final shape that you need for a protein. Think of level 3 as kind of like an entry requirement to be a protein, right? If you're an amino acid, right? And you have big aspirations one day to be a protein, okay? And you want to enroll in the protein army, okay? What is this? You need to, be as large as level 3. But you can also be level 4, which is an even cooler protein. Level 4 is kind of like the coolest, like top dogs at the military, okay? They're like the coolest amino acids in the protein army, okay? Now let me show you what level 4 is. Now we call level 4 a quaternary structure, okay? Now I know it kind of sounds epic, like come on bro, this is like one of the coolest sounding names in biology. And the main way this differs from the last one is that not only is it bigger, obviously, but the thing is with these is that they contain multiple polypeptide chains. The last one was only a single polypeptide chain, but so long, right? Like a long train. Now this one contains multiple polypeptide chains. Now the entry requirement to be a quaternary structure is that you need at least 2 polypeptide chains. If you have more than that, then cool, you're him. And a good example of that would be hemoglobin in your blood, right? In your red blood cells, you have hemoglobin, which is what carries oxygen and carbon dioxide, right? And this protein is so large, right? It's a quaternary structure, and it contains 4 different polypeptide chains bonded together. It's like, it's crazy, okay? And this is something you'll learn in a later topic, but just understand that when you have a quaternary structure, right, you have multiple polypeptide chains bonded together, and it is usually to serve a massive, crazy function. Or like monoclonal antibodies. And these guys have 4 polypeptide chains, and their structure is adapted to carry bacteria. It's pretty cool. And of course, these different polypeptide chains are held together by bonds that we've just spoken about. So, same concept, but not too crazy, not too difficult. Just learn that we start off with level 1, primary structure, and then we progress on to level 2 as we add more amino acids, which is the secondary structure, where it can either be an alpha helix or a beta-pleated sheet. And then you add more amino acids, it folds even more, and then becomes a tertiary structure where you have a different mix and combination of alpha helixes there, beta-pleated sheet there. And then, sometimes, a protein could fold even more and become like the big dog, right? The quaternary structure, where it has multiple different polypeptide chains bonded together. Pretty cool, pretty awesome. Now let's go over the biochemical test to actually find out if a solution contains a protein. We call this the biuret test, okay? I don't know why people say biuret, you're just weird, but this is a biuret test, okay? And it's pretty simple. First, you need to make the unknown solution alkaline by adding a base or an alkali, whatever. So here, just use sodium hydroxide, right? It's a great alkali, lovely, would recommend. Then you add copper sulfate, which is a nice, cool blue color, okay? You add it in, and if there is a protein present, it should turn purple or lilac, right? But if there is no protein present, it should just stay blue, okay? So pretty simple, pretty easy, just memorize that. And you'll be absolutely giggling in the exam. So yeah, that should be the end of the video then. If you enjoyed it, give it a like, subscribe to the video, all that bollocks. And I'll see you soon, bro.